Well, thank you, and I want to thank the organizers, Life Technologies, and Thermo Fisher Scientific for inviting me here today to present to you of the future of forensic science or forensic genetics. I need the slide. So we all have reasons why we do forensic genetics, and of course the motivations are victims, finding perpetrators, exonerating the innocent, as has already been mentioned, mass disasters, terrorist attacks. All these things present challenges to society, and we have to find ways to help investigate and solve those kinds of problems. So our goals are to identify the source of biological samples or associate them if we can't have an identification made, to exclude individuals, develop leads. We now have databases that help develop investigative leads, that help us solve more crimes. And we have all sorts of demands because with success, people expect more success. And so there's a greater demand on the forensic geneticists today than ever before to solve problems. So I can't talk about everything about the future. And usually predicting the future is not an easy job, nor are you held accountable for what you predict. But in this case, I think it's a lot easier because many of the things we're doing now are our future. So we don't have to look so far ahead to see where we're going and what we can do. So I thought I'd talk about maybe first thinking about what is forensic genetics, maybe about a little bit about sample collection, low copy typing, how that's impacting the way we think about statistical analyses or hypothesis-driven interpretations, rapid DNA, the ability to take something out of the laboratory and move it into the field for analysis, and obviously the next generation sequencing, which we'll find is really not the best way to describe what we're doing, but how that's going to help us do more things. So forensic genetics, molecular biology was probably embraced and implemented into forensic genetics before it was embraced into any other application field. And for more than 20 plus years, almost 30 years now, we've been using molecular biology to address the kinds of issues that we encounter. It's the gold standard in forensic science, and we love being told we are the gold standard of forensic science. But with that comes complacency. Once you're told you're great, you believe you're great, and you stop thinking about what can be done to do better. And so sometimes the problems we have, we don't talk about, and we don't really address them well. So we have to think about how we move forward and how there are still things to improve upon. Now, I've always looked at the definitions of forensic genetics and adhered to these that we've seen others explain, but most of the situations are we're trying to resolve conflict, a suspect and the government. The two sides making an argument or positions and each trying to prove the other's position to be correct. And that only subtly describes what we do, because we don't do that solely anymore. We now look at things like database searches. There's no suspect or victim there. You're looking for a suspect, and that's part of the job. Could be a molecular autopsy or determining ancestry of a missing person, human remains, where you don't have the same conflict in place. So we're expanding our roles, and we have to think of forensic genetics not just solving something in the courtroom, but also developing leads and gathering other information for other applications that may occur. Crime scene investigation, a lot of us are well aware of that, but I bring this up for one reason is we have to collect samples. If we don't collect the sample, we can't analyze it. If we don't collect the quantity of material that is at the crime scene, but we only collect a portion of it, we may not have enough to type something downstream. So collection becomes important to us. And there are lots of swabs. And what I see with almost all the manufacturers or developers of swabs, there are new polymers coming. There are new devices to be collected, because everybody recognizes if we can collect more at the crime scene, and more importantly, recover more from the collection device, we have a better chance of success because more DNA means an easier typing result. And just as an example, we've been working with one company called Diomics, who has its own polymer and collecting its own devices, and seeing that we are getting better recovery with the, I'd say probably the gold standard in there, the Copan swab. And we're getting better results for the same amount of material being collected in the devices. So we're going to see not just Diomics, but Copan and the others all making new devices that will improve the work so that we won't have as many low copy samples to start with. We'll have higher quality to begin, which would be good. And that brings us to low copy work. With success, you want to do more cases. And you want to take on more challenging cases because DNA can be useful in these, but it brings with it a lot of issues. So people have been looking at lower level samples and trying to recover. And we know that low level samples have baggage, a lot of issues that we have to think about. Allele dropout, allele drop-in. And yes, we're not going to be able to improve technically those things with the current technologies, but they have brought in something new that we'll start thinking about, and that's starting to drive more hypothesis-driven thinking process. Instead of just saying, I'm going to run something and do a calculation that, this is 1 in 8 million individuals, we're going to start framing things differently, starting to take in more information to get better assessments to express the uncertainty that exists with what we do. And when there's high confidence, that will be able to be given as well. So we're going to see going from binary models, which we were all advocates of at one time, to semi-continuous, to eventually continuous models, where we can deal with things of what is the dropout rate of a system, what is the drop-in rate, so contamination, loss of information can be addressed better so that we can deal with them with the uncertainty that exists when we go into these areas where there's more challenging samples. And we'll see where we start to think of all the possibilities. And I don't expect anybody to want to memorize this slide or anything, but just the idea that we can take any situation where there are uncertainty and put some value on it that allows us to better assess where the work's going in some probably logistic model that will be used to give us some better capabilities. One of the software out there we see a lot of promise is STR-Mix. Comes from the ESR and seems like it's a good tool. What's nice appealing about this tool, it's much more open. You know what's going into it, you know how it's being calculated, and I think we need those kind of tools. And so I predict the future won't be just any tool, but those that have this sort of open access capability so we understand what's going on. But there are issues, and I predict some of the future will be, now you have a tool, how do you populate the variables of those tools? It's not enough just to say, I choose a value. You have to do studies that reflect the reality of your samples. What is the dropout rate for a peak height of some value? You have to do validation studies, and they're going to be far more extensive than previous validation studies, and the community is going to have to come together to address how those will be done. Is it sound today to split something into 3 samples when it's already limited, amplify the 3, assess what's going on, or should you only amplify 1? We have more DNA into it that will approximate better what might be the contributions. Those will have to be reasoned out as you move forward because the 2 tools that allow for drop-in and drop-out say there's no reason to split them anymore. So as a community, this will be some of our future discussions I see will happen. We want to be able to do things faster, better, and cheaper. And that's always going to be, we heard from Mark just beforehand, everybody has limited resources, but yet the demands are great. So rapid DNA is another potential way of doing that. Now, rapid DNA is an interesting term, because rapid DNA to me means doing something fast. But the technology, rapid DNA technology, isn't necessarily doing something fast. It's doing something in a mobile fashion, being able to take the lab somewhere else. So we shouldn't confuse it from taking the standard approach to like direct amplification. Direct amplification is a rapid technique. You can take anything like identifier direct or whatever technology, take a cutting, drop it in, and get a result. That's rapid DNA. And in the laboratory, you can turn around things as fast as any of the devices. But the device is doing something different. So rapid DNA to us is being able to take all the different tools or techniques, putting them in a box, and taking it out and putting it in operation out in a field, instead of building a laboratory on site. So cost effective? Yes, very cost effective if you think about trying to build a laboratory. Easy to do, turnkey, turn it on, get a result on the other end. Lots of different companies have been developing. Some have gone to the wayside. Some have survived. Anybody can do it. I'll let you know that now because you don't need a lot of training to be able to stick a swab in a device and get a result. And you can get reasonably good profiles that can be used. And we see that there's a lot of potential applications in law enforcement, immigration at borders may be another thing. Human trafficking might be a really good application for this, where there's a real need to get quick answers on, is this child really the child of these parents, or are they a relative or not in the process? The military will be using it when they detain people in war zones and that. But the technology is also bringing us to the future of microfluidics. And we're going to see these technologies eventually one day go to the wayside for true microfluidic technologies that will allow us to use less reagents, less materials, less waste product to be able to get results. And that can also change our future. So I predict the rapid technology you see today may not be what we see in the future, but microfluidics will be part of our future. Now, we all have areas of interest, STRs, mitochondria, all SNPs for all the different applications, biodefense where it may be. And to do that, we need a greater set of tools, a bigger toolbox to be able to answer those markers and the applications that may arise. And we also have to think about the way we do things now. So what happens now? Almost all the countries now have databases, except maybe Italy, I think in Western Europe at this point, have databases. And all cases are driven by the database. You have a stain, you extract the DNA, and you run it on a kit that contains the core markers for your database, no matter what the case is. And we do that because we need to do that to be able to communicate the results from that to what may be in the database or share across countries the information, or cities or states as it may be. The problem with that is some cases can't be solved well by doing the core markers first, autosomal STRs. A victim who scratches her assailant and you take the scraping under the fingernails traditionally has mostly her DNA. And when you do the autosomal typing, you don't see the perpetrator's DNA. The first thing that should have been done was doing YSTRs. YSTRs provide tremendous amount of information, and if you're going to take one shot, you want to get the best information first, not the best searching opportunity. So we want to start thinking about how can we make better decisions in what we do as we move forward so we can solve more cases. And that's where next generation sequencing comes in. And I don't like the term next generation sequencing, because everything is next generation, right? So I've heard people call it now generation, so you can keep NGS. But we just go with the simple massively parallel sequencing, because that's what we're doing, and that's the technology. And I won't talk too much about it, other than all the markers that we have now that are in our databases do not have to be thrown out and start again, because we can sequence an STR and convert it back 99.9% of the time to the data that's in the database. So one thing that's nice about new technologies is when you don't have to throw away your existing data. So again, there'll be a greater embracing of a new technology like MPS, because we don't give up on the millions of profiles that have already been generated. Size doesn't matter anymore. And what I mean by that is when we do capillary electrophoresis, we had to tag them with fluorescent dyes, and then we had to space out the markers so we could know which one was which within a color. So you had to have a small amplicon, large amplicon, modifiers to move them around slightly. So the large amplicons, we started to compromise ourselves, because they're going to fail first in degraded samples and highly inhibited samples. So we already have better ways if we can bring them all down to more similar in size and smaller in size. So a new technology that allows us to amplify better and work with more degraded samples is going to be embraced. STRs, as I mentioned, will be one of the markers that can still be used. And if the technology allows us to do the core markers plus more, we can add in the markers that will actually do better than what we have now. or be additions to what we do now to be able to solve the problems. We also do SNPs. Now, most people would talk about SNPs. A few have done SNPs and have done quite well in identifying SNPs. But the community didn't say, oh, I love SNPs, I want to do it, because they're ingrained with STRs, even though SNPs could offer some advantages in certain applications. Now we can see that the certain applications can be achieved together at the same time that you're doing STRs, and that will be part of the toolbox in there. Now, the technology, in a lot of ways, is not so different than what we do now, and I don't like when people use the terms paradigm shift, quantum leaps, because this isn't a quantum leap. It's based on the same technology and knowledge we have already. You still have to extract DNA, and you still have to amplify DNA. And those are going to be the foundations that are going to have the greatest impact on the outcome of the result. So sequencing, to me, is just another detector. Yes, it has its own issues and things in there, but it's just another step in a process. And maybe it's more like forensic genetics typing on steroids. It gives you a lot more power, but it's not really a fundamental change in the way we think about things, although we'll have to figure out the noise and the details of the system. One of the systems we work with, and we'll see moving forward, is the PGM, a personal genome machine. It has some really nice features. It's unique, and I'm not going to talk about too much of the detail because others will speak about it. But the nice thing is the sequencing is rapid, so all the sample prep beforehand can feed a machine fairly quickly. And it's kind of ingenious because we don't use traditional fluorescent dyes and laser and optics to detect things. We're using computer technology to measure a change in pH. So whenever a nucleotide is incorporated into the growing strand, a proton is released, and that changes the pH. So in after we generate the library of DNA, which you'll get from others, put them into the wells on the chip, it will read them simultaneously with all the others. And so a very expensive pH meter, but a very effective pH meter in determining what are the potential sequences to generate what might be considered an ionogram that those who are familiar with can read very quickly what is the actual sequence that has been generated. The other system that we work on is the Illumina system. The MySeq has a high throughput, 15 gigabases, more like the Proton in the ion torrent system. Same sort of thing, but does a different system. It generates the clones for the library prep and for sequencing on a flow cell in clusters through bridge technology, which I won't describe, but uses more traditional Sanger sequencing strategy to be able to detect the sequences in each of the clusters to determine what the sequence of each fragment is in the process. Bioinformatics will become critical. Now, we've always had bioinformatics, and most of you don't realize that when you get a CE result, there's a lot of processing of data that goes on behind the scene that no one is aware of because it's all done for you. Eventually, that's what we're going to do, and we're already talking to collaborations with our colleagues, Christophe and others, to grab tools so that everybody has them so that all the behind the scene will be processed, make it easier for the user. But bioinformatics is necessary. So it's a field unto itself, and it's unlikely that there will be bioinformaticians in every single forensic laboratory. In fact, I make a prediction that there won't be bioinformaticians in every laboratory because it's just not going to fit the system. So I thought, just as give you an idea of what goes on in sequencing, to appreciate the computer processing power is, I put on one page 5,000 nucleotides of the human genome. And I ask you, how many pages does that take to create one human genome sequence? And the Human Genome Project sought 6x coverage. That means, on average, 6 times you sequence each nucleotide. So how many pages do you need for 6x coverage with that? And your answer is a lot of pages, right? That's the answer, okay? Yes. So if you did it correctly, and you did 6x coverage, you would have to have 40 boxes, 5,000 pages per box, six boxes per pallet, so about 18 pallets of boxes of paper, one human genome. And what do we do in sequencing? We throw it into a shredder, and we shred them into tiny little pieces, all randomized, and we throw them into the machines, and they somehow read all those shreds and then say, oh, we're going to put it back together in some sort of cogent message, some story. That's not a trivial process. You think about it. Try and put together that many pages of paper and try to figure out what's going on. Can't be done without the use of tools. And then, not only are we going to put them back together, we're going to look at each nucleotide, say where there is a reliable base, an unreliable, each read, what's reliable, what's not reliable. We're then going to do both strands and decide if there's any bias, whether that has any impact or not. Thresholds, coverage, and all these things will come in there. And I said we're going to need software. And 2 tools seem to have been produced to answer the question. And both these tools are good, one developed by Christoph here and then one by my group, both taking fundamental changes from the way the bioinformaticians in the companies saw the problem. Because they saw the problem as making, for STRs, virtual allelic ladders and then aligning to the ladders. You will fail because the motifs vary, the ladders were there, it would be a tremendous problem. Both of us, slightly different but very similar approaches, said there's a better way. So the community is standing now producing the tools that will become available in the near term. STRs are going to be done, and there are different groups producing STRs. Similar sort of coverage comparable to the relative fluorescent units, so no different. They don't have the same meaning anymore. You're going to see high level and low level going from small to large. They're all going to be more close in size, so it'll be more about performance than about that. And then we're going to see within STR variation. There'll be SNPs, motifs in the repeats and then also in the flanking area that will increase the heterozygosity power in the process. And this is consistent with the work, and I say the first work done by Oberacher et al. From Walter's group did the sequencing by Sanger, a very difficult process in those days, would have been trivial today, and mass spec to identify variants that existed. So we knew certain loci would have these variants. Why would we be interested in this and not just look for STRs that have more alleles and higher zygosity? STRs that have more alleles will have higher zygosity on average, but the spread between the smallest allele and the largest becomes greater. The greater the spread, the less efficient is the PCR, and we get preferential amplification. So we want to have markers that have high heterozygosity with the hidden variation because the spread of the alleles will be less and the amplification efficiency will be good. So now the markers feed back to more efficient PCR. So there's a whole process of embracing new things that can make us do what we're doing better. We can separate out alleles. I did it pictorially so you don't have to look at the sequence. This is a homozygote where we have both the alleles with different sequence, so it's truly a heterozygote. But I also marked in the stutter because the stutter is carrying the variant as well. And we have to know how much and how often that occurs because now we can start thinking about deconvolving mixtures better than the way we are doing it now to reduce the uncertainty. This is a great example, and I'll just show it as a picture. 2 different alleles. The red is now the stutter from each of those, the plus and the minus stutter. So now we can quantify the amount of stutter and actually do better in our stutter studies to lead to this, a mixture in which we have 2 people who are 11-12 heterozygotes. We can actually quantify the 12 contributors because they have a SNP that separates them, but not the 11. But since we did the 12, and now we know what the amount is from the 12 from the major contributor, we can make a better prediction that there are 2 11s, even though the sequence is there. Again, reducing down the number of possibilities, increasing our uncertainty, or decreasing our uncertainty, increasing our confidence in our result by this, but also getting in the stutter for the minor contributor so that we can do better in the possibilities that are around. So that's the future. Mitochondria, I'm not going to talk too much about it because I know Walter will talk more about it, but mitochondria is a tool. In some places, they're not using it. It's a tremendous tool that has a lot of value in a lot of cases, and especially the most challenging of cases, but it's expensive. And I look at some of the commercial entities, they charge somewhere between $1,000 and $3,000 a sample. It's a good business to get into because you can make a lot of money, but it's a lot of work. Heteroplasmy, having multiple species of very similar sequences, often during the homopolymer stretches that have downstream effects because of the alignment. Sanger sequencing sequences everything at once. So all the out-of-sync sequences get jumbled and makes it hard. Next generation sequencing, MPS, sequences each fragment individually. So we no longer have the same problem with the heteroplasmy as we had before. And again, other tools that we'll try to incorporate into this major toolbox that as a group we'll do, to take the current sequencing alignments and realign them to ensure that we're all doing it in the same fashion. So with a heuristic model, we can do better in the process. And population studies become easy. We, in a matter of short time, one individual did 283 whole genomes. That would have taken years under Sanger sequencing. And we're able to do that in a joint project of our group and Walter's group, and sorted through all the data fairly quickly. So now the tools and capabilities, the databasing as well, and we get a lot of information. We get, this is a circle plot where the outer circle is the coverage across the genome. The middle circle is the variance in reference to the Cambridge standard. So the small sites are variants that occur rarely. The long ones are the common ones in most of the population and differ from the reference. But one thing that's very interesting is 75% of the total variation for the mitochondrial genome was outside HV1 and HV2. H1 and HV2 have a lot of variation in a small region, but there's a tremendous amount of variation that can help you outside. So this is another area we can do that. Heteroplasmy, this is a study we did showing heteroplasmy. We found 4 length variants. One of them we did not see by Sanger sequencing, and we could tell you what each one was. Unfortunately, the tool at the time didn't allow us to quantify it well, but we do now there's 4 there. So the next tool will be to help quantify that and get better information. Bones, these are samples of bones run by Sanger and that, and exactly the same results between them. And then the next step is small amplicons going all the way around the genome to work for degraded DNA, because we're not going to be able to work with long PCR as we do with reference samples for degraded DNA. So we're going to see that as part of the future for our process. And so just quickly, some applications. We haven't heard anybody talk about doing it in casework yet because there's a lot of things we have to think about in validation. But there are studies we can do that can tell us whether or not it'll work. And we got contacted by the Historical Society in a state called South Dakota in the US, which has a city called Deadwood, very famous for the history of the US. During the gold rush, it established in the 1870s. And of course, wasn't the best of times. People died. They had a cemetery, and 2 years after the cemetery was established, decided to move it to another site and move the bodies. And then 100 years or 40 years later, they were building a retaining wall, and they found some remains. And they said, whoops, we did not move one of those remains. And because it's historical, they wanted more information. And now we have a tool, and we said, well, what can we do? So the first thing we did is we took the panel, we took the Y SNPs from the Ion Torrance HID panel, and they have 34 upper clade SNPs, allow us to analyze it, and lo and behold, it comes out with R1b, which is the most common haplogroup for Western Europe. So this fellow is very consistent with being from Western Europe. We then did the ancestry SNPs, and you can see depending on the panel of SNPs and also the reference populations, you get Western Europe, but slightly different, whether it's the Ken-Kid SNPs or the Selden SNPs, as might be expected in the process. And we did phenotype, and I have to say he's a very attractive, red-haired, brown-eyed guy, and I used to be a red-haired, still brown-eyed guy. So, so we can put a little bit more about the individual in history, which gives more of a personal reality to these people who are interested in historical work. Now, we also did some blind studies at a meeting we had in the US where they sent out 12 samples and said, what can you do about it? So we said, okay, what can we do with it? So we took our identity SNPs, the ancestry SNPs, the Y SNPs, and STRs and mitochondria, and said, okay, we'll figure it out. And we noticed that 3 individuals had the same haplogroup, and in fact, the exact same whole genome sequence for mitochondria. Said, maybe they're related. Very likely if they had the same sequence, but we didn't know the relationship. We just know that there's some maternal lineage there. But with the other markers, we were easy to tell what was the relationship. And whenever you're doing a study that's blind, you shouldn't put the last 4 individuals in the sequential order, blind samples. It makes it easy to do the study. But we were able to figure that out and have a very high likelihood ratio supporting this pedigree versus unrelated individuals in the 10 to the 40th range or something like that. So overkill. But I wanted to bring up a point about biogeography does not necessarily mean bioancestry. And the genetics we get may not be where the person originates from, ancestry-wise. And so we have to be careful and understand it in context and what are the markers as we move forward. So if you take the relationships of the grandfather and grandmother and where their bioancestry is, we see that the grandmother comes from somewhere in Asia. The father comes probably from Western Africa. The child, who didn't come from any of those places when she was born in there, looks to come from somewhere in the Middle East. Probably wasn't from the Middle East. It's, again, the choice of a limited set of markers and 2 parents and understanding that it's going to give us information, but we have to look at it in context of where it is as we move forward. Slightly different with each one. But we also had one individual who declared themselves of Central Caribbean origin. So as soon as I think of Central Caribbean, I think of someone of African and phenotype. And when we looked at it, they didn't fit an African phenotype. Afterward, we got some phenotypic information from the donors. They were fair skin, light eyes. Their parents came from France and Spain. They just happened to be now in Cuba. So again, as we start to think about admixed populations, mobile populations, we have to be careful what it is. In the last minute and a half, if I have that, other areas, identical twins. Anybody ever have a case where one is arrested, they do a DNA type on it, it matches the evidence, he says, oh, my brother has the same type, and lo and behold, brother has the same type. There are now possibilities. This work was done by Eurofins out of Germany on using where they sequenced both twins, whole genome sequencing, and looked for the somatic mutations between them to demonstrate that they could differentiate them. And then you could then type the evidence with traditional Sanger sequencing to be able to sort out which one is the donor of the sample. This is not just a research study. There's a case going on in the United States now that will go to court in the next month to enter in this kind of evidence to identify twins. So now, challenges we had before. I mentioned molecular autopsy. We know that also forensic identification, there has been work. Low copy DNA is hard. Humans leave very little DNA when they touch, most of the time, objects. But we have 10 times more microbes in and on our body than we have human cells. And if you swab the skin, you actually get 100 to 1 to 1,000 to 1 microbes per skin cell. So there's a lot more of some other DNA we're leaving behind that is likely to be unique with each of us as well and might improve our ability to analyze touch samples and also for biodefense. But in the end, we're going to have to get the research right and put it into practice, and that will be obviously the next step of the process. So in the end, we're going to have to deal with all the noise, the quality, the thresholds, the specific information in bioinformatics, and validation, and most importantly, education of the forensic scientists, but also the community of stakeholders in the process. So with that, I hope to give you a little bit of, we've come a long way, and we keep doing more, and we're getting better and better and being able to do a lot more than we were before, and we're not done. There'll always be more that'll be coming. With that, I'd like to thank all the individuals who have contributed to this work, particularly Thermo Fisher Scientific for allowing us to be here today, and thank all of you. Thank you.